So now that we've gone over some of the basics of molecular biology, in particular the principles of hybridization, probes, linkers, and molecular biology reactions catalyzed by enzymes, we can begin to delve into sequencing technologies and how we sequence DNA in this day and age. I do have to start briefly explaining the state of the DNA that it's in when we extract it from various sources, and then it'll go into specific sequencing technologies. So first, just sort of as an aside, you can use it as a party factoid if you want to impress your friends. How much does a human genome actually weigh? So I'll just tell you, we could derive it. If you want the exercise, you can look it up and derive it yourself. But I will just tell you that one human genome, let's say the one from your mom, weighs 3 picograms. That means that in 1 nanogram, 1/1000th of a gram, you have approximately 300 genomes. And I'm mentioning that number because a nanogram is still a relatively easily quantifiable amount of DNA. But if a nanogram only has 300 copies in it, then the downstream manipulations better be very efficient to retain most of that complexity. Otherwise, if you go down to, say, just a single copy of the genome, that's not enough to give you every place in the genome because, of course, it's statistically distributed how you sample it. So a nanogram is an important one to remember. Nanogram, about 300 copies of a genome, which also means that a microgram has about 300,000 copies of a genome, and a microgram is a nice amount of DNA that people really like to work with. Microgram is a good amount. More is better, but microgram is pretty good. So there is redundancy in our preparative amounts of genomic DNA. That's the main thing I wanted to get across. Okay, now as far as extraction of DNA goes, we get DNA out of solid tissue, of course, and in practice, you grind up the tissue, sometimes under liquid nitrogen so that it's nice and powdery, and then you put in some proteases, things that digest proteins, and then you precipitate the DNA out. Blood and cells are sources, and of course, white blood cells are cells, so cultured cells or white blood cells. As I mentioned before, cell-free DNA can be a source, but cell-free DNA is degraded to very short pieces and oftentimes present in very, very low quantities, sort of those kinds of nanogram quantities I was mentioning. And then there's also fixed DNA that comes from pathology samples. So when people prepare pathology specimens to look at the cells and see what the tumor looks like, it is fixed with a reagent called formaldehyde. And formaldehyde basically cross-links a whole bunch, but the main effect as far as our application here is concerned is that formaldehyde breaks DNA. And so these formalin-fixed, paraffin-embedded samples contain DNA, but the DNA is kind of in bad shape. Also quite short, 150, 200 base pairs, sometimes 500 base pairs. So depending on what you need to do, you are either stuck with really bad DNA, or you can get out of blood, you can get very good long DNA. But the larger point is that we cannot get DNA such that we can read the DNA from one end of a chromosome to the other end of the chromosome. It's 300 million base pairs, or in the case of chromosome 1, or 250 million base pairs. And just the physical manipulation of breaking the cell, extracting DNA and whatnot, it breaks down the DNA to shorter pieces. If you're really, really good in the lab and very, very careful, you can get DNA sizes of up to 100, 200 kilobases, 100,000, 200,000 bases. If you're not that careful, your DNA is probably more like 20,000 bases, 30,000 bases. And again, if it comes from one of those compromised kinds of sources, it's very short, 300, 400 bases. So that's the state of DNA after extraction. And quantification of DNA is usually performed either visually on a so-called gel. You run the DNA out in an electral gradient, and then you stain it, and you see sort of a smear. Or by more sophisticated methodologies, and I will draw this out real quick, where here's the amount of DNA, and here's the size. And there's a particular separation procedure that can be used at the same time that the DNA is labeled with a fluorescent dye. It's a dye that doesn't break the DNA. It goes in and positions itself between the bases. That's called an intercalator. Remember, we talked about intercalators earlier when we were talking about mutagens. So this is a reagent that slips into between the bases, and it fluoresces when you shine a laser on it. And so you can separate the DNA by size, and then depending on at which size you have how much signal, you can see something like, perhaps something like this, where most of the DNA is at size, let's say 1 kb, and here at size 10 kb, there's less DNA, and then it peters out. You have no DNA that's at 100 kb size. So these kinds of profiles tell you the quality of the DNA, and under the curve is the quantity of DNA. So that's a very typical kind of way to quantify DNA, just the raw genomic DNA that you've just extracted. Okay. So let's build some libraries from the DNA. And the purpose of building these libraries, and I will explain what a library is in a moment, the purpose of these libraries is to prepare the raw genomic DNA that you've just extracted to be read by the molecular biology and the instruments that do the DNA sequencing. And this is an important point. DNA sequencing doesn't happen on just raw DNA, at least not yet. There may be methods in the works where you can just literally drop a bit of DNA into a reagent chamber, and then it just sequences that DNA without any prior manipulation. But in the meantime, we're stuck with methodologies that require preparing the DNA such that it can be sequenced. And that preparation is called library building. So depending on exactly what you would like to do, the library building process is different. If you want to sequence a whole genome, you have two options, which we'll explain in a moment. If you only want to focus on the coding sequences, because again, those are the interpretable parts of the genome, the currently best interpretable parts of the genome, and you want to save money, then you do something that's called an exome, where you pluck out the regions of the genome that contain protein coding sequence. And you do that, as you might imagine, by hybridization. Or if it's even more limited than that, you have a so-called targeted panel, and that is done by PCR. So we're going to go over that. And then there is a procedure called RNA-seq. Again, this is not for genotyping or for DNA sequence assessment. This RNA-seq is done usually for expression analysis, because RNA is a phenotype. Remember, it's not a genotype. So let's address these one at a time and start with whole genome, because in some ways the whole genome bit is the easiest to explain. Let's start with fragments, or in the jargon, frag libraries. And in fact, I've already explained this, because what we have is fragmented DNA. And by the way, let's say that we did a very nice DNA prep that's, base 50 kb average size, okay, really beautiful DNA. Well, we have to trash it. We have to break it down to about 500 base pair pieces. So our genome is in 500 base pair pieces. Of course, this follows a size distribution because the breaks are random, so not everything is 500 bases. This is just the mean. There's lots of smaller pieces and a few larger pieces. And because we have reasonably large quantities, a few nanograms perhaps, we actually have several thousand copies of the genome. So let's say that, just for just to have a number, let's say that we have 10,000 copies of the genome. Okay, so we've really in our tube we really have oversampled the genome. Of course, we're not going to sequence each one of these pieces. We're going to sequence a smaller subset. So, well, let's build a library. And here, the procedure is pretty simple. First of all, when DNA comes out of an extraction procedure, the ends are ragged, and we have to polish the ends. So we're going to throw in a DNA polymerase with some DNTPs, and we're going to make sure that the ends are really at the same spot. And here's one last base pair. There's no base, no single base hanging off. Okay, it's really nice and beautiful. Okay, that's called end polishing. And of course, you have to imagine that this is done on all the billions of bits, right? All the billions of bits that are in my tube. I'm just drawing you one of the billions. Then as an aside, ligations work better if you actually do have an overhang. And so what we're going to do is we're going to make an A overhang that we're going to do by primary extension. And so there's a single A overhang. And then we're going to take a chemically synthesized linker that has a known sequence. And I'm simplifying here. These things are actually quite fancy and can be really engineered in very interesting ways. But I'm going to skip over that. And this linker here is synthesized such that, of course, it has a T here, a T overhang. All right. And so now what I can do is I can linker this up. And now I have a library where all of my genomic DNA pieces have handles, and all of these sequences are the same. So there's actually a trick, and I'm going to, again, this gets into the weeds a little bit, but there is a really beautiful trick by which it turns out that you can make it so the sequences are actually, on the two sides, are actually different. So this is this sequence here is different from, these are called Y adapters. This sequence here is different from this sequence. And so see what I've done here is I have the 5' end of either one of the molecules has the purple sequence, and the 3' end of either one of the molecules has the black sequence. And now what I've done is I have a library. After one round of PCR, I'm going to do one round of PCR here real quick. Okay, I have a library that has that has this black sequence on one end and a purple sequence on another. So I actually have different sequences on both of these ends. And just to be clear, let me just change this color here. I'm going to make them purple and red. Okay. So all of my DNA molecules now have two sides, and I can address them separately with these known sequences. That's called a frag library. So hold that thought. We're going to be sequencing the inserts here using linker sequences that are on both sides later on when I explain the sequencing process itself. Let's build another type of library really quick. Let's say that I have much longer DNA, perhaps 5,000 base pairs. And so these are long pieces of DNA. And I would like to, let me back up for a moment, okay? Long DNA is hard to sequence. There are technologies that sequence long DNA, but the short read technologies basically require making libraries of short fragments. Let's put it that way. I can't make a library out of this long fragment for the for the particular technologies that are the most widely used ones. And so sometimes there are some applications where I would like to know the sequence of the ends of these long molecules because the connectivity here provides information. And by some magic tricks that involve circularization of the molecule with a linker in between, I can make a library that is, again, that has two separately addressable ends, but where this piece here is actually a disjunct piece that was put together. So this is this guy, and this is that guy. And then with bioinformatic approaches, I can actually disentangle this. So in other words, what I've just done is I've built a mate pair library, that's what they're called. And mate pair libraries have the nice property that the sequences that I'm going to be determining here, I know are actually considerably further apart and give me extra information in ways that I can explain later. Further apart give extra information, and that has certain advantages for detection of structural variation in particular. So these are called mate pair or jumping libraries. In the end, though, this is the library that I've made, and you can see how that's really, it's the same structure as the fragment library that I just explained, only in the fragment library I have one contiguous piece of DNA from an original, and here I have 2 bits that I put together. So this is pretty straightforward, right? And I think that's partly why whole human genome sequencing will win out over these more complicated approaches that I'm going to explain in a moment here. So let's do an exome. And just to, I shouldn't have erased it. Let me just make the point once again. The goal of this library building is always to get you this construct here, this particular molecule, where this is the interesting piece, and these are the handles by which you manipulate it. So when I make an exome, that's my goal as well, because that's what goes on to the sequencer. How do I make an exome? I look at my reference genome and I determine that these sequences are coding sequences. So that's gene A, and over here is gene B, and here is gene C. So these are all the coding sequences. Now, what I'm going to do is extract DNA and trash it so that it's nice and broken up. And of course, I have lots and lots of copies of this. And I have, in the meantime, synthesized an array that has probes that match all the exons. Remember, I have the sequence of the reference genome. I know where the exons are. I can make oligonucleotides of 50 base pairs or so that match exactly the sequence of this exon. And I'm going to mix these, which, by the way, have a little handle, usually. I'm going to mix that, hybridize it, and then I'm going to pull them down by a handle. And now, by the handle, and now what I have is I have the DNA that matches these pieces, and all the other DNA has floated away. And then I'm going to put linkers on that DNA, and now I have an exome fraction. Now, in reality, there's always a little bit of noise. You know, some sequences come down that shouldn't come down. Other exons behave poorly, etc. But in principle, most of the protein coding space of the human genome can be addressed in such a way. And remember, only about 1.5% of the genome is in protein coding exons. So in principle, I could sequence all of these having a cost of 1.5% of a whole genome. In reality, this procedure costs something and so forth. And in the end, you basically sequence about 10% of the genome. These with very high specificity, and then there's a little bit of noise around. So So it gives me, approximately a 5-fold cost reduction or so if I only want to see the exons. And that's the reason why we jump through those hoops. Okay, that was an exome. Now let's say that I'm interested not in all of protein coding space, but for my particular type of cancer, let's say, we know that only a handful of genes is usually involved, and I want to reduce the costs even further. So how do I do that? Instead of going for the whole exome, I make PCR primers that flank the exons of those genes I'm interested in. And that's called targeting. So PCR primers, and remember PCR primers are highly specific to the specific sequences. And PCR allows me to generate preparative amounts of material, preparative amounts of DNA. So basically, I just run the PCR, and I can do this in parallel because the primers don't talk to one another. I can do this for all 300 genes I'm interested in. And so I get lots and lots of PCR products on this exon, lots and lots of PCR products here, lots here, etc. And in the end, I throw that all together, and I build myself, again, a library that I can sequence. So this is a so-called targeted panel. The targeted piece of it comes from the fact that I'm targeting the exons by these flanking PCR primers. And panel simply comes from the idea that it's a panel of genes. It's not all of genes, it's a subset. Okay. So those are the 3 main types of libraries, 4 main types of libraries that people currently use. Fragment, the mate pair, which is this special case where I get information on longer fragments, exome, and targeted panel. That's DNA. Let me briefly go over RNA-seq. If you recall, RNA, again, is single-stranded, usually with a poly-A tail. I make a DNA copy by reverse transcription, and then by a process that uses a DNA polymerase that is called, the process is called second strand synthesis. I basically replace the RNA and now make a double-stranded piece of DNA. Here's my double-stranded piece of DNA. Because transcripts vary in size from relatively short, a few hundred base pairs, to can be quite long, a few kilobases, we actually break these to make them into smaller pieces. And now you know the story. We have short pieces, 500 base pairs or so, and we link them up. So out of my RNA, I've made a sequenceable library that contains only the expressed portions, only the transcripts, only the portions of the genome that are converted into transcript. So that's RNA-seq in a nutshell. Now, what I learned from this can be diverse, and I can understand expression levels, but I can also understand alternative splicing and various other aspects that are useful. So those are the main types of libraries, and they all end up like this. Barcoding. This is an aside, a technical aside that's actually really quite important. I've just built a library, and I've told you that the linkers here are all the same sequence across the entire library. Now, it turns out that I can have slightly different flavors of these. And let's say that this is, patient 1. And now patient 2 is going to get a slightly different flavor of this linker. Let me just make a fake sequence here. And let's say that this is a fake sequence here. But other than that, they look pretty much the same. Okay. And remember, I synthesized these, and it turns out that the vendors that synthesize these linkers have gotten the idea that one can put little barcodes in here, basically indices that uniquely identify this particular linker. Now, in reality, this thing is about 50 base pairs long, okay? And the barcode is not just 3 bases, it's probably about 12 bases, or it can be even longer. But basically, every sample can therefore be tagged with a unique barcode that can be later sequenced in the same machine run that sequences the interesting part here. Okay, so you sequence this, but you also, in a separate reaction, but at the same time, basically, sequence this. And then you know that this sequence comes from this patient because it's this barcode. And this sequence here comes from this patient because it's that barcode. So separate sequencing reactions during the same machine run will allow you to disentangle who it comes from. And basically what this does is it allows you to pool the sequences, pool the libraries from different sequences, from different patients, and sequence them at the same time, because you can later then identify who, on the basis of the barcode, which sequence came from which patient. And it's done simply for cost reduction purposes. The new machines that are out there now have such enormous throughput that first of all, you would never sequence a single targeted panel, because you would be wasting a lot of sequencing reagents that way. Because targeted panels are so small, there's so little sequence. So you multiplex targeted panels, you even multiplex exomes. And nowadays, the machines are so good that you can actually even multiplex whole genomes, because a single machine run, a lane in a machine, gives you so much data that it's even overkill for a whole genome. So that's why this barcoding is really very useful and quite important. Okay, that was an aside, but of practical importance. Okay. One other aspect of DNA sequencing that I've already alluded to is the following. We build libraries with DNA fragments that are perhaps, 500 base pairs long. When we do sequencing, we actually only sequence up to a certain point that's determined by the specific chemistry of the machine and the reagents that are used. And through some molecular biology trickery, we can actually sequence both ends. Okay, that means that there is sort of an unsequenced bit in between. And the reason I'm mentioning this is that it highlights the distinction between the fragment, which is really the template that we sequence, or the original piece of DNA that came from our DNA prep. The fragment is different from the sequence. And if you follow the jargon these days, you will hear people talk about short reads and long reads. So this would be, let's say, 150 base pairs. That's a very typical read length for the current Illumina machines in particular. And so that's called short read. But you see, the fragment in principle could be much bigger than this short read, right? It's just that it isn't, because the particular sample prep that the Illumina machines and related approaches use, they can't deal with longer fragments. So short read in current use is almost equivalent with short fragment, but you see, fragment is not the same as read, right? Read is what we read off the fragment. There are approaches that are quote-unquote long read approaches, and of course, if it's a long read approach, the fragment also has to be long. So here's a long fragment, let's say that's 10 kb, and there are approaches that can sequence that whole 10 kb. Okay. Now, why wouldn't you always do that? I mean, longer contiguous information is clearly better than these short pieces that we always sequence. And the reason is quite simple. There are no mechanisms at this point in time where you can take a DNA preparation that has long fragments. Let's make them longer. Let's make them 20 kb. We take a DNA preparation that has long fragments and make quantities of these long fragments that are preparative that you can then sequence on a sequencer that reads through. The only way you can read long fragments is literally by single molecule approaches. So the technologies that allow you to read long reads off long fragments have high error rates because they work off single molecules and not clonally amplified molecules. And what that means, I will show you briefly. So here's basically the summary of long reads versus long fragments. So there are some approaches that use a single DNA fragment as a sequencing template. Pacific Biosciences and Oxford Nanopore are currently the ones that are in the market. It's challenging to do this long fragment bit to keep these single molecules stable during the sequencing. And the signal-to-noise ratio is bad, well, not so good, because we're sequencing a single molecule. We're not sequencing several molecules several identical molecules in parallel. And the cost is also higher. So if you can't do this, or if it's too expensive, or if the signal-to-noise ratio isn't good enough, then the next best solution is this clonal amplification of short single molecules. And I'll show you a cartoon in a moment. The initial single molecule from a library is used only briefly as a template to amplify more of itself. And then you have thousands of identical molecules and they boost signal. But because you have to amplify, you can't have a long fragment. It has to be a short fragment. That's the catch-22 in all of this. And so for these clonal amplification approaches, there are two different methods. There is the Illumina approach, which is the dominant one currently, that amplifies these single molecules that are immobilized on the surface. And there's also an emulsion PCR, and I will go over those in a moment. So remember, we've built a library. And as I've explained before, the library is a massively complex mix of initially individual unique fragments with common handles at the end, right? So common handles at the end, and then literally billions of molecules with the samples of the genome in short pieces. So a library can be PCR amplified to make more of it, but actually so-called PCR-free libraries are preferable and they are available. They give you a more even sequence coverage, because PCR, as you might imagine, PCR can introduce biases. For example, PCR has a hard time getting through GC-rich regions. And so when you make a library and you PCR it, you usually lose highly GC-rich regions. It's only a small fraction of the genome, but you lose it. If you have a PCR-free library, you retain it. So those are preferable. And then in the sample prep, in other words, the step directly before the actual sequencing reaction, a relatively small sample of the library, which remember contains thousands of genomes, all in small bits, a relatively small sample of that, about 1 billion or so, are individually and separately amplified to give clusters of identical molecules. And here's the cartoon that shows this. So I have a flow cell surface to which probes are attached, covalently attached. These are oligos, about 20 bases long, or actually a little bit longer, I think. Anyway, they're covalently attached. And when you flow the library over it, the handles of the library hybridize with the covalently attached oligos here. So this is basically you're immobilizing individual single molecules. This is still when it was random. Now these are actually patterned, so they're in an array. But the point is that these things here are the handles that via Watson and Crick hybridization pull down individual denatured molecules. And they are sparse. And then there's a trick by which these get amplified to clusters. And this amplification step here makes identical molecules from that original starting molecule. Okay, so these are all identical within themselves. These ones here are different, but identical within themselves. Okay, this was one starting molecule, this was a different starting molecule, this was a different starting molecule, this was a different starting molecule. But we're making many identical copies of each of them. And now that we've made many identical copies of each of them, the actual sequencing reaction proceeds in lockstep. It proceeds the same in all of them, and that massively boosts the signal. So that's why these short fragment, short read approaches have a relatively low error rate, because you're assaying many, many identical molecules at the same time, and that gives a nice robust signal. And again, remember, each one of these is a different molecule, a different DNA sequence. And an analogous process that I'm not going to go into too deeply here involves something called emulsion PCR. Basically, the point is that, again, we have linkers, in this case on a bead, a single DNA molecules in there. It allows you to amplify, much like you had on the surface, in this emulsion on this bead. It allows you to amplify that particular clonal molecule. So you have this bead that contains that molecule, this bead that contains that molecule, and you then purify the beads and spread them on the flow cell. And so it's basically equivalent. In the end, you have a flow cell where you have a bead with identical molecules, also with handles, versus a flow cell where you have the identical molecules immobilized on the surface. So two different approaches. This one is the ion torrent approach. This one is the Illumina approach, as far as current technologies is concerned. For various reasons, this appears to be the dominant one. It does give longer sequences and has higher throughput at this point. Okay, now as far as the scale goes, and I'm just picturing the beads here, you can have the same concept with the Illumina flow cell where you have the individual clusters of molecules. Same idea. These are tiny. The places where these molecules are amplified are tiny. They're very, very densely packed. And basically, the newest machines that use these flow cells in their sequencing have 10 billion clusters, 10 billion of these little things per flow cell, which is, just a microscope slide size thing. So you have 10 billion clusters. Each of them contains a different clonally amplified DNA fragment. And from each of the clusters, you're reading 150 base pairs from one end and 150 base pairs from the other end. So you're reading about 300 base pairs for each of the 10 billion clusters. So that's 3 trillion bases. That's enormous. It's an enormous throughput. And so because it's so highly parallel, that ends up being the best pricing, at least for whole genome sequencing. There are also lower throughput machines. As you might imagine, this is a marketplace, and not everybody wants to buy the really super expensive machine that has this gigantic throughput. Some people, especially in genetic testing companies, some don't require that much throughput because they have targeted panels or exomes. And so there are other machines that have lower throughput, perhaps sometimes a little bit easier to use. The machine itself is less expensive. And overall, the cost is less, but of course, per base pair, the cost is higher because that's how they do it. Okay, so I'm going to skip over this. I'm going to explain how the sequencing reaction itself works in the flow cell. This is one of the many identical molecules in that same spot in the flow cell. This is linker sequence, and this is the DNA whose sequence we would like to determine. We anneal a primer, and now we're going to do primer extension one base at a time. So here we have our deoxynucleotides, and they are labeled. So there's a color for each of them. And we attach one base at a time. This is the G, so that means that there's a C here. If the G attaches, that means that that's a C. It's not something else. That's a C. And then we wash things away. And then we shine a laser on it, and it gives us the color here. And we record that as an image. And this happens, as I'll show you in a moment, this happens in a massively parallel way across all of the different clusters. And in this cluster, it happens with all of the identical molecules. So every identical molecule in this cluster gets that G, and so you get a nice, robust fluorescent signal. There is some very interesting chemical trickery here. So note that we had a whole bunch of these nucleotides here, but only one got incorporated. Well, we did that because the next one, which, there's a DNA polymerase that attaches this. So in principle, it should be able to just synthesize this in one fell swoop, right? The base is all around. But these are all chemically modified such that the 3' hydroxy, the OH, at which the next base would attach is actually blocked chemically. And so part of the process of removing the excess here after one is incorporated is to actually also remove the block, so that in the next cycle you can add, after the imaging, you can add the next one. And so it's a stepwise process. It is attach, wash away, image, remove the block, as well as the color. This shouldn't be colored anymore. This doesn't give a color right now. It's only this guy that gives a color. So you remove the fluorescent dye at the same time that you remove the block. And then you attach the next one and so forth. So one after another, 150 times on one end of the molecule. Then you turn around with some trickery. And you sequence the other end of the molecule. And so now you've generated 2 times 150 base pairs. This is an old slide. It still has the old Illumina name. Solexa was the original company that produced that. Solid is now Thermo Fisher that produced that. Basically, same idea. You have separately addressable lanes. So you can have 8 samples here, and there's actually another flow cell that has another 8 samples. Reagents are flowed in here. That goes out to waste, and after all of these different cycles that I was just explaining, there's an imaging step, and a microscope goes over and takes images at every place here so that the individual tiny little microscopic clusters can be seen. So one picture after another, one picture after another, and there's like 100 pictures that are taken at every cycle. And in the solid workflow, it's the same. So here's how that works. This is our flow cell, and I'm showing you 3 clusters. Okay, this cluster is it's the first cycle. This cluster has an A. This cluster here has a C in the first cycle, and this cluster has a T in the first cycle. And this is a cartoon simplification, of course. In the next cycle, this cluster has a C. This cluster has a G, and this cluster has a G, and this cluster also has a G. In the next cycle, that one has an A, this one has a G, this one has a C. And so this is the sequence that you're determining. So in the first cycle it was red, in the next cycle it was blue, and the cycle after that it was red again, so A, C, A. And then the next cluster has T, G, G, right? T, G, G. And the next one has C, G, C. And you just repeat this until number 150 or thereabouts. And you do this for all 10 billion clusters all at the same time. So the signal analysis of all of this is really quite complicated. You don't just get a base call out like this. There is a whole bunch of incredibly sophisticated bioinformatics and image processing that goes into all of this. And it's done, in these machines, it's done with microscopy. The one that I skipped over, the ion torrent, I should say, is a totally different mechanism. And it uses electrical current as a readout. So the strengths and weaknesses of these short fragment approaches, first of all, the amplification that's required to make many identical molecules on the flow cell means that we can only deal with short fragments, regardless of read length. If it requires amplification, we can only do short fragments. The pro is that these many identical molecules are all in sync, and they boost the signal. But the con is that no biological process, and these are biological processes, right? They use enzymes. They're chemical, but biology derived. No biological process is perfect, and so actually there's a possibility that the molecules actually dephase, where you one for some reason skipped the previous incorporation, and then it's sort of one behind. And you can see that in the raw signal, and there's a whole bunch of signal processing that corrects the dephasing of the individual molecules. And another aspect of it is that you have multiple chemical reactions, one base at a time. Now that's very highly controlled, and so that's an advantage. You really have full control of the entire process, but if you only have 99% efficiency, after 100 cycles, you're actually down to, I think it's approximately 30% or so of the molecules still working. And so it really requires that each individual process, each individual step has a very high success rate, because if it doesn't, the signal will peter out exponentially after just a handful of cycles. So in the end, what you get is a file that looks like this. This is the identifier for the particular sequence. There's another name here. And then I think these are 100 base pair sequences. So these are all, see, all of these clusters, these are all different clusters on that flow cell, and they generated these different sequences. So this bit here comes from over here in the genome, that bit there comes from over there in the genome, etc. And for the moment, you don't know where they came from. That requires the bioinformatics. But for now, you get these individual sequences The files they come in are called FASTQ files because they don't just have these sequences, they also have so-called quality scores. That's where the Q comes from. FAST is a historic name. Doesn't matter where it came from. But basically, for each one of these, you also have, for each position, for each base, a quality score. And the quality score is derived from, exactly what that raw signal looked like. And it's designed to give you an estimate of the probability of correctness. And I want to just very briefly explain the quality scores because they're slightly counterintuitive. I'll just do it by example, okay? A quality score of 10, Q10, means 10% probability that it's wrong. So you could say you sort of have 90% confidence. Okay, but hold on, 10, that's not it. Okay, 20 means that it has a 1% probability of wrong. So you have 99% confidence. So 30, you see where this is going? 0.1% wrong, 99.9% confidence. So in other words, this is a log scale, and this is 10 to the negative 1. This is 10 to the negative 2. This is 10 to the negative 3. Okay, so now you see 1, 2, 3, 10 to the negative 1, negative 2, negative 3. So this is the negative log of the error probability, error P, times 10. Okay, negative log 3 times 10 is 30. So a quality score of 30 is generally considered pretty good. Quality score of 10, not so good. So you don't trust that base. So if that base here, if that G has a quality score of 10, you better have many, many reads that give you the same G in the same position before you really trust it. But you don't trust it right away. The other point to note is that, and I should have a slide for this. I forgot. The other point to note is that at least for the Illumina chemistry, error rates tend to be relatively high at the beginning of the read, and then they sort of flatten out to be reasonably low, and then they start back up again towards the end of the read. So the quality score distribution that you will generally see in these Illumina reads is sort of low, 10, 20, so forth, and then 30, 30, 30, perhaps higher, and then back to 10, 20, and less. So the middle of the main portion of the read, a little bit towards the front, is usually the highest quality. And the ends are not so good, oftentimes. Okay, that was quality scores. Let's talk about other types of sequencing approaches that are currently in the marketplace. In particular, long reads and short reads on long fragments. So we were just talking about short fragments the whole time, right? Because the short fragments are necessary. You can't do the sample prep, the cluster amplification on long molecules. It has to be short molecules. So those short fragments. What if we are interested in long fragments? And as I noted earlier, these are single molecule approaches, these long reads, okay? And the two ones that are currently in the marketplace, there's a third one that's probably coming sometime soon, use extremely different methodologies to give you long sequencing reads from long fragments. And they are amazing, amazing rocket science. So I want to talk about these two briefly so that you get sort of a flavor of the technology that's behind those. Also, parenthetically, they are still more expensive than the dominant Illumina technology, and they also have higher error rates, and that is because they sequence single molecules. And the error per base, if it's just a single molecule, it's just simply higher. So let me explain Pacific Biosciences for a moment. And again, this is grossly simplified. Basically, they have a whole bunch of these nano wells. And in the well sits a DNA polymerase, a single molecule DNA polymerase. And actually, I've attached the DNA polymerase to the long DNA fragment before I put it in the well. That's a technical detail, but basically, the DNA polymerase has a fragment attached to it, and the reason it can do that is because there's a linker here. Again, it's the handle idea. This linker here is really good at hooking up to a DNA polymerase. But the DNA polymerase can't do anything with this long single-stranded piece of DNA because it doesn't have nucleotides. You can only put in a nucleotide and begin to sequence if you have nucleotides floating around. So what do we have? We have nucleotides floating around in the space above the flow cell. Detection comes from here, by the way, and it's done by a laser. And I'll show you in a moment how that works. So you have a nucleotide floating around, and that might be a G. Maybe I'll just call it a G. Okay. And a T. And these nucleotides are at very, very low concentration. And they're in such low concentration so that it takes less than a millisecond, but a millisecond is a long time. It takes a millisecond for things to go in and out, okay, out of this tiny little well. But if the right one goes in, and let's say that the right one here is an A, the polymerase will grab it and make molecular contacts with this A. And that process takes long enough that the polymerase will grab it. And if it's the right one, it incorporates it as the next base in the growing strand that's templated off the crick. So it's making the Watson off the crick, right? And that takes a while. So the covalent binding, the catalysis takes a while. And so this A hangs out in this well much longer than diffusion in and out. And so you get a relatively long signal. That's the fluorescent signal of the A label. And then when it's incorporated, it actually loses the dye, and so the signal goes back down. And then let's say the next base is a T. And all the while, random nucleotides float in, but they don't actually hang out very long. But the T is incorporated, and it makes a big signal, etc. So depending on the sequence, you get, again, sort of a color signal here, and it's all done by single molecule. So there's not many molecules that are identical. It's a single DNA molecule, and it's a single A and a single T, etc., that gets incorporated. And it happens in real time. You just let it sit, and you shine a laser on it with a certain frequency, and then you read that off. Now, it looks really good here in practice. This is quite a noisy signal, and there's a whole bunch of, again, image processing, if you will, that happens before you can actually make a sequence out of it. But in the end, depending on how long the DNA was, and what the quality was, and basically the stochasticity of the individual molecule approach, in the end, you can get reads that are up to several kb, and the mean read length is nowadays probably around 10 kb or maybe even 20 or so. So really nice long reads. But they do have high error rates because, as you might imagine, a single fluorescent molecule gives us a single sort of burst of light, and that can be misread or it can be missed entirely. And so the error rates of this approach is on the order of 10%. So that's PacBio, single molecule sequencing by these nanowells, where it's one base incorporated at a time. The second approach, the nanopore approach, is completely different. Briefly, what we have is a electrical. So this is the flow cell. This is the reagents, and this is electrical detection. And this is a membrane here. This is a synthetic bilayer membrane, and we've inserted a protein that's called hemolysin. This protein is a nasty one. It, as you might imagine from the name, hemolysin, it lyses blood cells. It's a bacterial protein that kills blood cells by making a hole in the membrane. And the fact that it makes holes in membranes has been exploited. So it has a channel. I'm really bad at drawing these. But basically here, it has a channel. And of course it's three-dimensional. So this channel allows ion flow. And if you put an electric current across this from here to there, then you can measure the electric current, the ion flow. Long story short, if you put DNA in here and thread DNA through, the exact amount of electric current depends on the base pair sequence of approximately 6 bases of DNA in here. Okay, so what you get is similar to the PacBio, except that the data are completely different. You get this signal of electricity as the DNA passes through, and depending on exactly the signature here, you can infer the base sequence. It's really utterly remarkable that this is possible. And of course, the protein itself is highly engineered, and the methodology here took years to develop. But similar to PacBio, you can get very long reads of individual DNA molecules out of this particular procedure. I won't go into too much detail about this piece here, because I will briefly describe it in the summary slide in the next one. These are long fragment, long read approaches that require single molecule manipulation. There is a very interesting technology that is relatively new that actually retains the information of long fragments, but utilizes short read sequencing, specifically Illumina sequencing, in such a way, and I will just draw this out really quick, in such a way that these different long fragments produce reads in the end that have a barcode that is different amongst the different individual long fragments. It's done by microfluidic partitioning. And so basically, if you just look at this cartoon for a moment, I have a barcode sequence that says that this read and this read and this read and this read and this read came from the same long fragment. So I'm retaining long-range information, even though I don't do single molecule sequencing, but instead what I'm doing is Illumina sequencing of these barcoded short fragments. So I know these are the black ones. I know that these all came from one long fragment. These are the purple ones. I know that they came from another long fragment. And these are the red ones. They all came from yet another long fragment. And so these guys go together, these guys go together, these guys together. So that allows me to utilize long fragments, retaining long-range information, but having the superior error rate and low cost of the Illumina sequencing technology. This is very much researchy at this point, but I think that it will make its way into genetic testing at some point as well. So, in conclusion, short reads are here to stay for whole genome sequencing. Until long reads improve error rates and are cost competitive per high confidence base, in effect per amount of information that I get out. As of now, the error rates are still relatively high and the cost is prohibitive, so it's a marketplace. The short read approaches win. They will also stay for whole genome sequencing while short reads on long fragments, this procedure that I just explained over there, while that improves, because it's still relatively new and the bioinformatics hasn't quite caught up to it yet. But that's very promising methodology. So short reads are here to stay for the foreseeable future. And they are here to stay forever for short fragment applications. If you have cell-free DNA that you've isolated from the maternal blood, that's in 150 base pair pieces, there's simply no reason to use a long read approach. There may be a reason to use a single molecule approach, but a long read approach is not necessary. So short reads are here to stay for those applications for which we don't need long reads. For whole genome, yes, eventually long reads will prevail. But for counting applications or fragmented DNA, short reads will stay. And so I think that we're entering a phase in genetic testing where the science has come so far that genetic testing companies are adopting these kinds of approaches now, and because of the long timelines that we're looking at, we'll be retaining those approaches for the foreseeable future, maybe for the next decade or perhaps longer, actually.